Thanks for your interest in the MCAT Intensive Review. I'm Dr. Ogan Gurel. I want to take a moment to share with you the background behind the course, and in particular some of my educational experiences and the influence of great professors and teachers that I've had that have made the course ultimately what it is today. So I want to start with my Harvard education. I got a biochemical sciences degree, cum laude, and I worked with a number of professors at Harvard which were extremely influential. For example, Professor William von During, Professor of Organic Chemistry, who was a major contributor to understanding aromatic properties of molecules, and also a major contributor to carbene chemistry. He taught a very, very tough organic chemistry course, and a lot of students were quite in trepidation of taking his course. And I realized that the difficulty, if you will, was the mathematics and the quantum mechanical base of organic chemistry, and the application of rigorous analytical thought, and not just memorizing reaction mechanisms. And that was a tremendous lesson, and when we talk about achieving understanding for the MCAT and not just purely memorizing, that's part of his legacy transmitted from him through me to you. Another professor was Professor Guido D Higgins, Professor of Biochemistry. He was my biochemistry professor, and he's made major contributions in understanding the structure and function of membrane proteins, in particular the sodium potassium ATPase and the insulin receptor. And a great professor, and I really remember those times in the biochemistry course fondly. Professor Guidotti was also joined with Professor Jack Strominger, teaching the biochemistry course, and he has made major contributions in understanding the major histocompatibility proteins, and their role in antigen processing and CD4 T4 cell function. Another professor I took cell biology freshman year was Daniel Branton. Some of you may be familiar with him. Professor Branton was the discoverer of the freeze fracture method in electron microscopy that provided substantial evidence of the lipid bilayer model of biological molecules. It was a real pleasure to be taught by a professor whose work was featured in almost all the major, and is still featured in almost all the major cell biology texts of the time. He's also worked in clathrin-coated pits and receptor-mediated endocytosis, and I had the privilege of working in his laboratory one summer on clathrin and related proteins. More recently, he's worked on nanotechnology applied to the sequencing of single DNA molecules. Another professor very influential was Professor Cynthia Friend, who was then an assistant professor of chemistry and is now chairman of the Department of Chemistry, and I'm not at all surprised. She was a master teacher, and really learned a lot from her. Her research has been involved in the surface and interface chemistry, and surfaces are very important in biology, membranes, in cells and tissues, etc. And so her interdisciplinary work is very important. I did my senior thesis work with Martin Karplus, and he has been a monumental figure in physical chemistry, in the fields of chemical dynamics, in NMR spectroscopy. There are even equations named after him. And most notably, the molecular dynamics simulations of the motions of macromolecules. My senior thesis in his laboratory was on the solution phase structure of DNA as determined by constrained NOE, nuclear Overhauser effect constraints, on molecular dynamics simulations of DNA. I also worked with Dr. Michael Weiss, now at Case Western Reserve, on that project. Between college and medical school, I worked at the Institut Lalangevin in Grenoble with a marvelous professor, Professor Joe Zakai, who was a pioneer in the application of neutron diffraction techniques. There's a great neutron reactor at the ILL to biological systems. And in fact, he and his team discovered the inside-out concept of membrane proteins, whereby soluble proteins have the hydrophobic core in the center, and membrane proteins have the hydrophobic surface on the outside. And the experimental evidence for that was provided through neutron diffraction studies. Worked in his lab to be one of the first to, actually the first to correctly determine the positions of two of the helices of a membrane protein, bacteriorhodopsin, in the sequence of bacteriorhodopsin, the structural map and the sequence. We identified helices A and B correctly, and it was one of the first glimpses into the detailed structure of a membrane protein back in the late 80s, and we published a paper of that. So that was a great experience and great impact. In medical school, Dr. Chen, who is now the chair of bioengineering at the University of California, San Diego, a monumental figure in bioengineering, and was my physiology teacher and very influential, and a great gentleman as well. Professor Eric Kandel, who you may know as the 2000 recipient of the Nobel Prize in Physiology or Medicine for studies in understanding the molecular basis of short-term memory formation. I was also his student in the neuroscience course, which is a famous course. There's a whole big book by that name read by students worldwide, which is really the definitive textbook on neurosciences. And he was a professor of that course and really influenced me through this breathtaking presentation of the neurosciences over the course of that year. Dr. Andrew France, my advisor and also the Associate Dean of Admissions at the Columbia University College of Physicians, was a great influence on me. In fact, he's been an influence on thousands of students, Columbia graduates and currently practicing physicians, of course, and he has had a profound impact on American medicine. He was a renowned endocrinologist in his own right. In his laboratory was the first to identify prolactin, and he developed a bioassay around prolactin, understanding its normal physiology as well as its role in pituitary diseases. Other important physicians that I worked with, especially in the clinical years at PNS, College of Physicians and Surgeons, were Dr. John Lerb, Dr. Kai Salakati, who discovered or invented the oral rehydration therapy for cholera, literally saving millions of lives, potentially worldwide. Dr. Glenda Garvey, unfortunately now passed away, but beloved memory as a master clinician teacher. Dr. Linda Lewis, who was the Dean of Students at that time. Dr. Donald Landry, who's a leader in the understanding of artificial enzymes, and many others, and I apologize if I missed some of them. Many surgeons I've worked with have included Dr. Robert Solomon, chairman of neurosurgery and a pioneer in advanced cerebrovascular surgical techniques, and Dr. Michael Treat, a profoundly brilliant surgeon who's invented a lot of interesting new technologies and approaches to general surgery. I'm also indebted to Dr. Peter Wyden, who was my psychiatry preceptor there and a world leader in the pharmacodynamics and pharmacokinetics of antipsychotic medications. He's now here in Chicago, the head of the psychosis program at the University of Illinois, Chicago. And finally, Dr. John Truman of pediatrics, who's now professor of clinical pediatrics at Columbia, was a marvelous influence on me. He would have afternoon teas in his office with the medical students, and we'd discuss cases and really learn a lot from him and from each other. Finally, Professor Wayne Hendrickson was my graduate studies advisor while at Columbia, and he is a world-renowned X-ray crystallographer and structural biologist. He's developed some of the most powerful techniques in macromolecular structure determination, including the multi-wavelength anomalous dispersion technique, and he's really been at the forefront of advancing all those structures of proteins and DNA that you are aware of now. He's also, at his lab, has worked on significant projects such as the GP120-CD4 complexes, which have led insight into the understanding of HIV pathogenesis. And then at Mass General Hospital in surgical internship, there have been a number of physicians that I've had the privilege of working with. Ultimately, one of the interesting concepts is that, as they say, you learn from your patients. And I've had the privilege of learning from hundreds of patients as a surgical intern and resident at Mass General Hospital. And it's a two-way street because in ancient Greek, the word doctor means teacher. And the role of a doctor is to teach patients how to avoid disease, prevent disease, help them about their disease, learn about disease so they can overcome that difficulty. And then, of course, you learn from your patients as well. But obviously, I've worked with some great physicians at the Mass General, and these include Dr. David Ratner, a master surgeon and a tremendous innovator in minimally invasive techniques. Dr. Matthew Hutter, who taught me that one never gives up. Dr. Kenneth Tanabe in surgical oncology. Dr. James Allen, Dr. Dean Donahue in thoracic surgery. And finally, Dr. Joe Grosella and Dr. Frank McGovern in urology. It's been a privilege to work with them, and I especially feel glad that I have the opportunity to transmit in some way to you some of the great teachings that they have provided to me. Thank you very much.